Bismillahirrahmanirrahim. Today, inshallah, we'll talk about the DNA, one of the most fascinating topics in biology altogether. DNA, as you know, is the genetic code, and if you think about it, it's quite a powerful molecule. In other words, it is a code like 0, 1, 1, 0, like zeros and ones that make up the code of a computer program. It holds the code that gives the commands for the color of your eyes, color of your hair, whether your hair will be curly or straight, your height perhaps, and perhaps also human intelligence. So if you think about it, it is quite a powerful molecule. The DNA is. It is the genetic material. When we say we get half of a genetic material from a mom and half from a dad, we mean half of the genetic code we get from individual parents. And it is quite a remarkable protein, and for me, it's also exceptionally beautiful in the way Allah has made it. The genetic code, the DNA, the structure of DNA was elucidated or discovered by Watson and Crick, James Watson and Francis Crick, as you see over here in this picture. Watson, in fact, wrote a autobiographical sketch of his times, of these exciting times, and they had published their famous discovery in 1953 in the journal Nature, and is one of the most important scientific discoveries of the last century. And the book itself, that's the one that was written by James Watson, so here is quite an important and beautiful read, and easy to read also if you ever get an opportunity. This particular model that you see over here is in fact in the British Museum. Also, if you get a chance to go to London and visit it, and in my mind is also one of the most beautiful things to see, and remarkable how beautiful the structure is, and how what it took it for them to discover its nature. So we're going to try to appreciate the structure, and today we're going to kind of get the big picture of what the DNA is, and subsequently we're going to elaborate on the DNA. So as you recall, the karyotype is the lineup of all the chromosomes. And if you recall, we have 23 pairs of chromosomes, or 46 chromosomes altogether, and including the X and Y chromosome, the 23rd pair. And of these pairs, one is from dad and the other one is from mom. Which one is which, we don't know here, obviously. I'm just guessing. And similarly, so we have 23 pairs of homologous chromosomes. Now, each of this chromosome is a one big long strand of DNA, and this is of course coiled up. So if you were to look at a cell, and if you were to look at the nuclei of a cell, that's where you would find the DNA. And when the DNA is all uncoiled up, it's like a spaghetti. That's called chromatin, of course. And when it's all coiled up in a nice sausage shape type thing, that's a chromosome. But the thread that makes up the spaghetti, so to speak, is the actual DNA, of course, and that kind of looks like this, the double helix. DNA, if you recall, is one of the 4 macromolecules that we find in our body. From a previous study, we had classified there are 4 macromolecules, or big molecules, that you find in living things. And these included, of course, proteins. And proteins were made of individual subunits. The basic subunit of proteins was the amino acid. That's the monomer that made proteins. And this is an example of alanine, if you recall the structure of protein. And then if you string together a bunch of amino acids, you get a polymer, a polypeptide. And if you string to a whole bunch of them, you get a protein. So amino acids were the building blocks of proteins. And similarly, we talked about basic sugar units, and they, for example, over here is 6-member sugar ring. These are the basic blocks, building blocks of of sugar molecules, and here's a fatty acid. So fatty acids, sugars, and amino acids and giving proteins constitute the major other macromolecules. And the building block of DNA is the nucleotide. So just like the amino acid is the building block of protein, it's the brick that builds the building of proteins. The nucleotide, that's what it's called, the nucleotide, is the building block of DNA and RNA. Now, RNA is also made of nucleotides. Now, if you string together a whole bunch of nucleotides, you get DNA. Now, if you wanted to describe the big picture structure of the DNA, the word DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid. If it doesn't flow smoothly from your tongue, you got to practice this, because you can't graduate from biology class unless you're able to articulate or enunciate deoxyribonucleic acid without difficulty. So DNA stands for deoxyribonucleic acid. It sounds kind of cool, right? Deoxyribonucleic acid. If you wanted to describe the big picture appearance of the DNA, you describe it as a double helix. In fact, that's the title of James Watson's book, The Double Helix. It's a double helix because there are two helical structure. That's like a helix, right? There are two helical structures that kind of swim around each other like this. They kind of embrace each other in terms like that. So that's a double helix. There are two strands that are helical in nature. They're twisted about themselves like a rope. So that's what the double helix is. So if you wanted to describe DNA with certain adjectives, the first adjectives you would be using, you call it a double helix. Now, the double helix, see, it has an outside and an inside. So it has like this outside, right, backbone, right? So and then it has this inside part of it, right? Okay. So this backbone, the outside part, is a sugar phosphate backbone. That's what that's called, the sugar phosphate backbone. That's kind of the lingo or the buzzword to describe a DNA molecule, a sugar phosphate backbone. In other words, this ribbon, which forms like the backbone of the DNA molecule, is made of sugar molecules. If you recall sugar molecules, right? They are either a 6-membered ring like this, or they are a 5-membered ring like this, right? So in fact, this is going to be a 5-membered ring sugar. Along with, attached to it would be a phosphate group that you will see. So, so if you were to describe this DNA molecule again, you would describe it as a double helix that has a sugar phosphate backbone, and in between this sugar phosphate backbone are base pairs. In other words, this is a base and this is a base, and they're always paired. See, in this case, purple and blue and are paired, so to speak. And I'm not quite sure this is accurate, but there's in the middle of it are base pairs. Okay. So that's something that you have to understand. So if you wanted to describe the DNA molecule, you would describe it as a double helical structure with a sugar phosphate backbone, which means sugar phosphate on the outside, and base pairs on the inside. So deoxyribonucleic acid DNA is a double helical structure, that is, it has two strands. It's a double helix, meaning two helical strands with a sugar phosphate backbone and base pairs on the inside. Now, here are the base pairs. Now, when Watson and Crick were trying to discover the structure of DNA, believe it or not, for almost near the climax of that particular story, some people still didn't think DNA was particularly important. For the longest time, people thought proteins were the genetic material. People knew about genes, but they didn't quite know what genes were proteins or genes were DNA or whatever. The reason is because protein structure is actually more complicated than this elegant, beautiful, simple structure of the double helix. Just to consider, there are like 23 amino acids. But in terms of bases that are in DNA, there are only 4 bases in DNA. these bases, the one that go inside. And in the DNA, the 4 bases are adenine, thymine, guanine, and cytosine. These form the rungs of this ladder, as you can you describe. So the rungs of the ladder are made of these base pairs: adenine, thymine, guanine, and cytosine. And adenine always pairs with thymine. Guanine always pairs with cytosine. And here, as you can see, this is cytosine, and this is guanine over here. This is adenine. This is thymine over here, and this looks like guanine and cytosine over here, and this looks like adenine and thymine over here, and guanine and cytosine, and thymine and adenine. So they're always pairs. So they're like best buddies. They never go. They always go in pairs like that. Okay, so a cytosine will never pair unless there's a mutation or something like that with thymine or something like that, or with adenine. Cytosine will always only pair with guanine. Okay, so these are base pairs. They always come together. So if the DNA could be likened to a ladder, the rungs of the ladder would be these base pairs, and on the outside is a sugar phosphate backbone, as you can see over here. So these are the base pairs, and that's the sugar phosphate backbone. Now, there are different ways to portray DNA. These are space-filling models that you see here on the right, and the other ones were more molecular models that you see over there. So there are different ways to describe the shapes, visually to appreciate the shapes, and this is a 3-dimensional representation of DNA. And then if you look closely at these base pairs, here's adenine. They look like very fancy looking molecules over here with the rings over here, adenine and thymine and guanine and cytosine. If you observe then, it's quite apparent that this thymine, for example, and cytosine, they have only one ring over here, whereas adenine and guanine have 2 rings, right? It's 1 ring, 2 ring, 1 ring, 2 ring, like that. So that's immediately kind of, at least to me at least, catches my eye, that they're different in the sense that one has 1 ring and the other one has 2 rings. But these are some structures of those bases. Now, this one shows the sugar phosphate backbone, and it shows those base pairs and how those base pairs are always linked. Adenine will always combine with thymine, and guanine will always combine with cytosine. So this is how the DNA structure is. Now, as I suggested, there are different ways to represent the DNA. So here's a cell. It looks like it's in dividing mood and has made these chromosomes, already duplicated this DNA. So if you look at a chromosome and if you unravel it, and you get like this, and then you will notice that if you were to unzip this ladder, so to speak, and the guanine will be combined with cytosine, and thymine with adenine, and cytosine with guanine, as you can see. In other words, these base pairs are complementary. In other words, if you know what the next base, if this was C, you can say that on the other side there would have to be a G. Cytosine would be guanine, cytosine would be guanine, thymine would be adenine, and etc. like that. So the two sides of the DNA, the 2 strands of the DNA, this is the 1 strand over here, and this is the 1 strand, they have complementary base pairs. This complementary base pair, and it is the sequence of these 4 nucleotides that determines what the gene is. It's like for computers, you have 11011110011, like that. It's a binary code for computers. There's only one of 2 choices, but the DNA genetic code has 4 letters, so there's greater variability, and it is this sequence of the nucleotide that determines actually what the code is. And hopefully, inshallah, when we are done with discussion of these topics in the next few chapters, if you appreciate the big picture and how the DNA actually determines, for example, your eye color, then it makes them quite beautiful, because DNA is a code that codes for certain proteins, because all proteins are the blueprint for the structure of the protein is the DNA. So DNA codes for the protein, and it's the protein that's in your eye that gives your eye color. So ultimately it's the DNA that determines a person's eye color, for example. So now if you look at the nucleotide in detail, is that there are 3 parts to it. This is the building block of the DNA, and the nucleotide has 3 parts. Okay, it has a sugar base, right? Sugar here in the middle, and it's kind of like the sugar's it's got 2 arms over here. On one side is the phosphate group. That's a phosphorus with a bunch of oxygen attached. And the other side is here on the other arm, so to speak, is the nitrogenous base. So there are 3 components of a nucleotide, the building block of DNA. The 3 components of the nucleotide is the sugar molecule. The sugar molecule, in this case, it's going to be a 5-member sugar with the oxygen right there. And on the 2 arms over here, is the one side is going to be the phosphate group, and the other side is going to be nitrogenous base. Okay. Now, I just want to suggest all the nucleotides are same in this way. All the nucleotides are same. They have the same sugar group and the same phosphate group. What's different between the adenine, thymine, guanine, and cytosine is the nitrogenous base. So this part of the molecule, the sugar and the phosphate, does not change between different nucleotides. It is the nitrogenous base that changes between different nucleotides. So here's, for example, an example of a nucleotide. So this is a molecular structure here of guanine. Okay, the guanine of the 4 nucleotides in DNA. Now, a guanine is a base with double ring structure, like over here. Okay, and this is the sugar molecule with the oxygen, as you recall. Now, the carbon in the sugar molecules are numbered. This is called 1', 2', 3', 4', and 5'. Those are the carbons. So if you wanted to talk about, okay, point to the carbon number 1, you would say it's right there. Carbon number 4, right there. Carbon number 5, right there. So the carbon molecules are named in the sugar molecule with prime. Now, why are they called primes? Okay, that's because they're nitrogen carbon atoms over here, and these 2 are numbered. So these, I don't know exactly how they're numbered. They're like 1, 2, 3, 4, 5. They're not prime. And to distinguish this carbon from this carbon and this carbon, whatever, so you use a prime as a distinction between these 2 different carbons. So this is that's why it's called these primes are used, as I understand. So this is carbon number 1. Now, of the carbons that in the sugar, the ones that are labeled here, they are labeled because they're important, the labeling. The 2 most important carbons are this one, the number 3, the one that has this hydroxyl group attached, and this one, number 5, which has the phosphate group. So this is the phosphate. Now, phosphate molecule is just a phosphorus attached, surrounded by 4 oxygen molecules, okay? And as an adenosine triphosphate, ATP, has this very similar structure to a nucleotide right here, understand? So, this 3' carbon is very important because it has this very important hydroxyl group. And this is very important group because it's involved in constructing a DNA. And the other very important carbon that's important is this one, 5' group. So the 3' carbon and the 5' carbon in the sugar molecule are super duper very important, and they're over here. So it's important to appreciate that, and for number 1 here. So now, as I suggested, all the nucleotides are the same over here. They don't change. What changes from one nucleotide to the other one is this part of it. That's the nitrogenous base. Now, why is it called the nitrogenous base? Okay. Well, if you just look at the darn molecule over there, you'll see there's a whole bunch of nitrogen there. Nitrogen, 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 and nitrogen. And well, why won't you call it nitrogenous base? Because it has lots of nitrogen. There's no nitrogen over here, right? Do you see one? No, I don't see one. So there's no nitrogen. So these bases have lots of nitrogen, and that's why they're called nitrogenous bases. Okay. So this sugar has a phosphate molecule on one side attached to it and this nitrogenous base. So altogether, this whole molecule here is called a nucleotide. Now, there's like a whole bunch of terminology that is associated with nucleotides, okay? And if, for example, if you just wanted to talk about that part of it, that's called a nucleoside. So, etc. But if you just appreciate for now, for us, a more fundamental understanding of these things, this altogether, this makes a nucleotide. Now, if you wanted to change this to a adenine, you would get rid of this and put the adenine molecule over here. If you want to make this a cytosine, you would put this one over here. And then the thymine, you would put this group over here, all attached to carbon 1. So which one of these base is attached to the sugar molecule at the first carbon determines what it's going to be. I hope that's appreciable. Okay, so here we have it. Now, the 4 bases then are different. The 4 bases, the 4 nucleotides are different based on which base is attached, which nitrogenous base is attached to carbon number 1 over here. Okay, and the 4 bases in DNA are these 4 over here. Okay, now as I have suggested, there are 2 types of bases: the ones that have 2 rings and ones that have 1 ring. The 2-ring bases, they're called purines because they're pure beauty, I guess. I don't know. So I have a little mnemonic here. Purines have 2 rings. That rhymes somehow. Purines have 2 rings. Pyrimidines only have 1 ring. So adenine and guanine are purines, and thymine and cytosine are pyrimidines. So 2-ringed nitrogenous bases are called adenine, and single-ring nitrogenous bases are called pyrimidines. Now, if you look at these individual bases, there are only very slight variation between one and the other over here. Look at adenine and guanine, for example. The difference is right here, okay, and right here. So in guanine, it's like you take that one over there and you put it over here and put an oxygen in its place over there. So there are very, very subtle differences between these 2 bases. And over here, the basic skeleton is the same. The difference again is in cytosine, it's right here, and there's an extra carboxyl group over there, extra CH3 group over there. Okay, so they're very similar in structure, but sufficiently different. Okay, so the 2 purines are adenine and guanine. So purines have 2 rings, adenine and guanine, and the 2 pyrimidines are thymine and cytosine. Okay, now, so here's purines. Now, as we said, adenine always buddies with thymine, and guanine always buddies with cytosine. The way I remember is these are adenine is kind of like pointy straight lines, just like thymine, pointy straight line, whereas guanine is kind of circular, curvy kind of letter, and cytosine is curvy kind of. So guanine always goes with cytosine, adenine always goes with thymine. Now, if you notice though, okay, a 2-ringed base pairs with 1 ring, a 2-ring pairs with 1 ring. So altogether, there are 3 rings between the 2 of them, right? So adenine is a purine, it pairs with pyrimidine. Guanine is a purine, it pairs with pyrimidine. And that's why in the end, they're always like, altogether there are 3 rings in the middle. Regardless of how you see it, there are 3 rings in the middle. And that's why it's like a symmetrical width of this molecule, so to speak, right? Do you follow? And that's why, if you notice, they draw this longer than they draw this, because they're trying to suggest that's a 2-ring structure and that's a 1-ring structure, right? So the G, the symbol here is taller than the C to suggest the 1-ring versus 2-ring structure. So from what you learn from here is that purines, the 2-ring bases, always pair with 1-ring bases. So 1 purine and 1 pyrimidine. So in the rungs, in the rungs of the ladder of the DNA, at any particular rung, you will find 1 purine and 1 pyrimidine. One base that is has 2 rings, and the other base will have 1 ring. So at every single rung, you will have 2 different type of base pairs like this. Now, how are they attached to each other? The DNA is in the middle, is attached. The two strands of DNA are attached to each other by hydrogen bonds. Now these are weak bonds. Now these hydrogen bonds exist between the two bases on each side, so they're weak bonds. But if you have enough of these hydrogen bonds, like a Velcro, if you have a tiny Velcro, it's pretty easy to remove. If you have a long stretch of Velcro, good luck, because it's hard. So the more hydrogen bonds you have, the more stronger the strands of the DNA will be attached to each other, and there are lots of nucleotides. You follow? But the two strands of DNA are attracted to each other by hydrogen bonds that form between these base pairs on each side. They're not covalent bond, they're not ionic bond, but they're weaker hydrogen bonds. That's important because these two strands need to move apart from each other, right? They need to move apart from each other so that the DNA can replicate, DNA can make a copy of something, give it and make RNA, etc. So it allows unzipping of the two strands of DNA, and then when they're done, it comes right back. Okay, so that's how the two strands are attached to each other. So here we have purines and pyrimides again. Thymine is attached to adenine, one ring and two ring, and cytosine attaches with guanine, and they attach with hydrogen bondings. Now, the DNA, deoxyribonucleic acid, and RNA are related structures, but there are some critical differences. DNA is a double helical structure, like you see over here. RNA is only one strand, so RNA only has one strand, whereas DNA has two strands. RNA is always generated from DNA. RNA is like a photocopy of the DNA. But another critical difference between DNA and RNA is, whereas DNA has adenine, guanine, cytosine, and thymine, RNA has adenine, guanine, cytosine, and uracil. So this is new. So it's slightly different than thymine. The difference between uracil and thymine is just a little tiny group, but that's a sufficient difference. Okay, so you will not find thymine in RNA. So RNA is different from DNA in that it's one-stranded, and secondly, it does not have thymine. Instead of thymine, it has a slightly different nitrogenous base called uracil. Now this is all important basic knowledge that I hope you master very, very well. So this is the distinction between DNA and RNA. To help you remember, pure as gold, okay, pure as gold, purines are adenine and guanine, pure as gold. As I said, purines have two rings. So purines are adenine and guanine, and cut the pie, cytosine, uracil, thymine are pyrimidine. So if you want the mnemonics to remember, cytosine, uracil, thymine are pyrimidine, have one rings, and pure as gold, adenine and guanine are purines, pure as gold. So that you have to kind of know. So now look, we're going to construct the nucleotides. So you attach that bond over there. That's a nucleoside when you do that. And then when you attach a phosphate group to it, the whole thing is called a nucleotide. So in this picture that you're seeing over here, we're constructing a nucleotide when you take a base over there and attach it to a sugar. And that becomes a nucleoside, S-I-D-E. And when you attach a phosphate group to it on top of that, you get a nucleotide, okay? Now, oops, daisy. So I wanted to show something else to you here. Let me just say, sorry. Well, be patient with me for 1 second. Okay, constructing the nucleotide. Now, this is a nucleotide. Now, the difference between RNA and DNA is very small. Okay, deoxy means without without oxygen, hydroxyl group, and RNA we'll see is slightly different in sugar. Okay, we'll get to that in just a little bit. Now look at this. We said the two strands of the DNA are attached to each other because of the attraction between these base pairs. So here you see cytosine and guanine, and the hydrogen bonds that form between these two molecules. Hydrogen bonding over here, hydrogen bonding over here, and hydrogen bonding over here. Because oxygen and nitrogen are both polar atoms, so they attract each other. And here's a hydrogen bonding that occurs between thymine and adenine. Hydrogen bond over here, and hydrogen bonding over here. So these are the hydrogen bonds that form between those bases, okay? So again, purines and pyrimidines are always paired like you see over here. You see the bonds, hydrogen bondings. Now look, now how do base pairs connect with each other? How do the base pairs connect with each other to form this long strand of DNA? Well, here's adenine, cytosine, guanine, and thymine connecting to each other, okay? Now, what's really important to understand is the connection always occurs from what's called 5' to 3' direction. In other words, 5' is over here. New nucleotides are always added right here to the 3' hydroxyl group, to the 3' hydroxyl group, to 3' hydroxyl group. So the next nucleotide will come and attach to that oxygen right there, to that to the 3' carbon. So that's why that 3' business is so important, because the new nucleotides are, that's a 3. The new nucleotides are always added at the 3' carbon to that hydroxyl group over there. So that's very, very important to understand. Now, let me repeat that in slow motion, okay? New nucleotides are always added at the 3' end of the molecule, okay? So it's always over here. So now observe again. New one is added at the 3' end, okay? That's very important. In other words, DNA is always constructed from 5' to 3' direction. DNA is always constructed from 5' to 3' direction like that. And there's another thing that we need to appreciate about DNA molecule, in that the two strands of the DNA, the two sides of the ladder, okay, they are called anti-parallel. In other words, traffic is going in different directions. It's just like a road. On one side is 5' to 3', and the other side is 3' to 5'. What does that mean, you say? Well, I'll show you what that means. This is called anti-parallel. Okay, they're anti-parallel. What this means is this. Now, I have here, to demonstrate this to you, I have utilized one of the iconic heroes. He uses Yoda. So if you imagine Yoda sitting here in sugar, okay, he's over here. Yoda sitting here in sugar. It's a 3' molecule, 5' molecule. You can see Yoda's head is that way, right? Towards that. And here's another Yoda over here. Two different base pairs, okay? Now, what this means, the two strands on one side is all the heads are facing this way. On the other side, it's like the other way. The heads are facing this way. See, on this side, the oxygen molecules over here, and then coming back over here, the oxygen molecules are over here. So it'd be like upside down, right? So the two strands are anti-parallel. Look at this side. The 5' is over there, the 3' is over here. 5' is over there, 3' is over here. 5' is over there, 3' is over here. On this side, 5' is over there, 3' is over here. It's going this way. 5' is over there, 3' is over there. 5' is over there, 3' is over there. So they are anti-parallel. This arrangement is called anti-parallel arrangement of DNA. So it's not like this. The two strands are not like this. This would be a parallel arrangement, okay? But it's more like this, okay? One side 5' to 3', the other side 5' to 3' in the other direction. This is called anti-parallel direction of the two strands of DNA. Now, if you look at it more high-tech, cool version over here, you can see, okay, the 5' carbon over here is over here, 3' is over there. Okay, 5' is over there and 3' is over here. 5' is over there and 3' is over here. On the other side, it's just backwards. Okay, 5' is over here. I'm gonna go dizzy over here. You know what I'm saying? 3' is over here. 5' is over there. 3' is over there. It's in the other direction. Okay. Now that's the way it is. We're almost finished. Think of this. Now here, you'll see how new strands are added to DNA. They're going to be added at right there at the 3' direction. Hydrogen bonds are going to be attracting those other sides like a glue, and those hydrogen bonds are going to just attract the right nucleotide. When that happens, okay, that phosphodiesterase bond that between the two is going to form. See, they're going to make a hydrogen bonds over here, right here. Come on over here, Velcro, and then over here you're going to form that bond. So that's how DNA is new DNA is formed. Now you also observe, my dear student, please observe how one strand almost like dictates what nucleotide is going to come in, right? Because they're always paired. So one strand acts as a almost like a proofreader of the other side. One determines the other one. As long as you have one side, the other side nucleotide sequence will be determined by that. Now look how beautiful it is also how the hydrogen bonds form, attracting that until that final strong bond is formed between 5' carbon hydroxyl group and this phosphate group over there. And also observing this beautiful rendition of somebody did how the two strands are anti-parallel. One is in this direction with the iodized head that way, and the other one in this direction with the iodized head the other way. And this is just another suggestion how this bond is formed. Look at this hydroxyl group, okay? I thought there was going to be another one. I guess there's not. Action, replay. Let's do this again. Okay, now watch this hydroxyl group. There we go. That's where it's going to be, okay? The phosphodiesterase bond. That's what that fancy bond is called. So here it shows if you wanted to duplicate DNA, you just unzip it and just attach new, and then you have two DNAs. And to me, I don't know about you, but I just find this fascinating. And you can, this is just different ways of looking at DNA. Now this beautiful picture here highlights another aspect of the structure, the big picture of the DNA. And that the DNA has what's called grooves, okay, with little valleys. And there's two grooves. There's a minor groove and there's a major groove. If you can see, look at the DNA structure. There looks like a big valley here and a small valley here, right? So it alternates between big valley and small valley. So the big valley, that's called the major groove, and the small valley is called the minor groove over here. You see the minor groove, major groove. So those are the major and minor groups of the DNA, which I think it's better highlighted in one of the earlier pictures which we have seen, like over here. Look at the major groove, right? And there's a minor groove. There's a minor groove, right? And there's a major groove. Minor groove, major groove. So the rungs are over here. Okay, major and minor grooves of DNA. The last thing we're going to talk about the structure of DNA is about these strands, okay? And that is, see, you can climb the stairs going this way, and you can climb the stairs going this way, okay? If you were to climb the stairs going this way, your left shoulder will always be towards over here. If you climb the stairs going this way, your right shoulder would be always towards over here, correct? Now you're still climbing, but in one way you're going like this, right? And the other way you're going like this. Now, is there a difference between that two types? Of course there's a difference between the two, okay? But you'll see, I hope. So which way is DNA, you'd say? Well, DNA is like this. So the two strands are both going like this. So the DNA is, it's twisted in this way, okay? Like that. It's the right hand. It's anti-clockwise twist. Okay. So here's a beautiful cartoon somebody did. As you can see from the model, the structure is basically a long, twisty, ropey, laddery type thingy. When it was first described, it was actually quite beautiful when it switched to description, 1953, in the Journal of Nature by Watson and Crick. Until next time, as-salatu was-salam ala sayyidina wa ala alihi wa sahbihi.